Imagine a world where the effects of aging on our brains could be reversed. Researchers at UC San Francisco have made a groundbreaking discovery that could change the way we understand brain aging. A protein known as FTL1 is at the heart of this issue. In studies involving aging mice, an excess of this protein was found to impair memory and disrupt the vital connections between neurons. However, when scientists intervened by blocking FTL1, they observed a remarkable transformation. The mice's brains began to function more like those of younger animals, and their performance on memory tests improved significantly. The hippocampus, a key player in aging. Aging has a particularly detrimental impact on the hippocampus, the brain region crucial for learning and memory. The researchers at UC San Francisco have identified FTL1 as a significant factor contributing to the decline associated with aging in this area. By examining the changes in genes and proteins within the hippocampus of mice over time, the team discovered that FTL1 was the only protein that consistently differed between younger and older mice, highlighting its importance in age-related cognitive decline. Mimicking aging in young brains, the findings were striking. Older mice exhibited elevated levels of FTL1, which correlated with a decrease in the connections between hippocampal neurons and a decline in memory performance. To further explore this, researchers artificially increased FTL1 levels in young mice, resulting in brain and behavioral changes that mirrored those of much older mice. In laboratory experiments, neurons that were engineered to produce high amounts of FTL1 developed abnormally, failing to grow complex, branching structures, and instead forming only simple single-armed extensions. Reversing memory loss by reducing FTL1. In a remarkable turn of events, when scientists reduced the levels of FTL1 in the hippocampus of older mice, the outcomes were transformative. The neurons in these mice began to form more connections, and their performance on memory tests improved significantly. Saul Valeda, Ph.D., who is the Associate Director of the UCSF Bakar Aging Research Institute and the senior author of the study published in Nature Aging, expressed his excitement. It is truly a reversal of impairments. It's much more than merely delaying or preventing symptoms. Metabolism, STL1, and future therapies. Interestingly, in older mice, FTL1 was also found to slow down cellular metabolism in the hippocampus. However, when the cells were treated with a compound that stimulates metabolism, these negative effects were mitigated. Valeda remains hopeful that this research could pave the way for new therapies aimed at counteracting the detrimental effects of FTL1 in the brain. He stated, "We're seeing more opportunities to alleviate the worst consequences of old age. It's a hopeful time to be working on the biology of aging. This research opens up exciting possibilities for future treatments that could enhance cognitive function in aging populations." What do you think about the potential of targeting proteins like FTL1 to combat age-related cognitive decline? Could this be the key to unlocking a more youthful brain? Share your thoughts in the comments.